So I have bad news and I have good news. The bad news is for those of you who came today hoping to see Congressman Holt, I'm not Congressman Holt, and I don't even hold a shadow compared to the congressman. I'm Larry Goldstein. I'm from UC San Diego, and I have the fortunate position of telling you the good news. The good news is that you're about to hear from a very distinguished scientist and clinician, Dr. Max Weisha, from the University of Michigan, where he is the founding director of their Comprehensive Cancer Center and a distinguished professor of oncology. His topic, as you'll see, is a new way of looking at cancer and thinking about how stem cells might be at the root of some kinds of cancers, a very exciting and important topic. Dr. Weisha was trained as an MD in the Department of Medicine at Stanford University, thus reinforcing something I've always known, which is that all good things come from California. He then moved on to Michigan after a stint at the National Cancer Institute, and he's been there, since 1980, and has a very distinguished record, including serving as chair of the board of the American Association of American Cancer Institutes and past chairman of the National Cancer Institute's National Cancer Center Support Review Committee. Most important to someone like me, and I hope to you, is that he has been an absolutely groundbreaking medical research scientist, taking advantage of federal funding that you all help provide for medical research to really begin changing the way we look at cancer, how it develops, and how it might ultimately be treated. And so with no further ado, Dr. Weisha.
Thank you very much. Is my microphone on? Can everybody hear me okay? That way I can just talk over here. So it's a pleasure for me to talk to you today, and I would also like to reiterate that none of the types of research I'm going to talk about today really wouldn't be possible without the support of the NIH. It's been absolutely critical. And I'm, as you heard, I'm a practicing clinician, a medical oncologist whose specialty is treating women with breast cancer. And I've been at this now 27 years, and I can tell you that there's never been such excitement now about some of the directions of research that we think are going to make a really fundamental difference. So I'm going to try to present a balanced picture, though, of why, on the one hand, we're so optimistic, and on the other hand, why has progress been so slow in cancer. And you'll see that we actually believe that some of the reasons behind this are locked in some of the secrets of stem cells. And I'm going to explain what I mean by this and tell you a little bit about, in general, about stem cells and then cancer and stem cells. And then I'll tell you about why embryonic stem cells are actually important for understanding what's going on in this research. And we'll open it up for questions then after that. Those of you who have not had the chance to come to Michigan, this is our Comprehensive Cancer Center, and it's a wonderful place to work. So I'm going to use breast cancer as a model for what I'm talking about today because, again, that's the area of my own research. But as you'll see, what we've discovered in breast cancer, we think actually applies to many other, if not all other cancers. Of course, cancer is a terrible problem in this country, and so is breast cancer. Over 180,000 new cases diagnosed this year, and over 40,000 women will die of breast cancer. I'm going to tell you, starting with the good news and then the not-so-good news, and then why the not-so-good news is going to be good news. So that sounds a little bit convoluted, but if you actually look at the bottom line, which are death rates from cancer, in the USA or in Britain, they actually went up every year until 1990, and then there was an inflection point, and every year the death rate from breast cancer has been coming down 2 to 3%. So that today a woman's chance of dying from breast cancer is about 25% less than it was in 1990. It's wonderful news. When you stop to look at that, you could say, well where have we made the progress? Half of that is due to early detection, mammography. And the other half is what we call adjuvant therapies, or treating very early stages of breast cancer, where we can reduce the recurrence rate in about half with hormone therapies or even chemotherapy, or more recently, what we call molecular targeted therapies, which I'll get to again in a few minutes. So that's kind of the good news. So why do I say the news isn't all good if we have a 25% reduction in death rate? Well, this is a magazine that's not at all a medical magazine. It was Fortune magazine. And this was about 2 or 3 years ago, and it generated a lot of controversy in the field. It was written by a gentleman, Cliff Leaf, who said, we're losing the war on cancer, and how are we going to win it? And he showed these kind of more grim statistics. And as opposed to looking at overall death rates, he looked at what happens to patients who already have advanced or metastatic disease. Have we really made progress against cancer despite all of the wonderful research that's going on? Has it really affected the lives of people with advanced cancers? And what he showed in this article was very sobering, that for the 4 leading cancers, prostate, breast, colorectal, and lung, once the cancer actually spreads to distant organs, the actual 5-year survival rates actually haven't changed very much. Now, it's changing a little bit in breast cancer now because of some of these targeted therapies, but it's still woefully inadequate. An interesting sidelight is the fellow who wrote this article, Cliff Leaf himself, is a cancer survivor. But he happened to have a kind of cancer, Hodgkin's disease, which is largely curable. And he was in a clinical trial at the NIH and was cured of his Hodgkin's disease. I found out later, I didn't know it, but I was the fellow who treated him when I was in my training right down the road at NIH. So it was actually an interesting discussion of why haven't we made more progress. And what I'm going to show you is that our recent research suggests that the reason that the progress hasn't been more rapid, at least one of the reasons, is we may have been targeting the wrong cells in cancer. And indeed, there are only a small populations of cells in cancer that we call cancer stem cells. And these are highly resistant to treatments. And if we're going to make real progress, we think we have to figure out much more clever, targeted ways to get rid of these cancer stem cells. Let me talk about the overall idea here, which others have termed to the cancer stem cell hypothesis. Now, the idea that cancer and stem cells are related is actually, in a way, a very old idea. Matter of fact, way before stem cells and embryonic stem cells were discovered, 100 years ago, was suggested that there's something like happens in cancers that resembles what goes on during development in embryos. But it was really only recently, over the last 5 to 10 years, that advances in biology and stem cell biology have really allowed us to examine this cancer stem cell hypothesis. There are really 2 parts to this, but they're separate, actually. The first concerns where do cancers come from in the first place. The conventional wisdom for the last 50 years has been that any cell in your body, if it got the right series of genetic mutations, would form a cancer. And that was, again, an existing dogma. The cancer stem cell hypothesis says that's not the case at all. There's a more limited set of cells that can actually turn cancerous. As a result of that, when cancers actually form, the second part of the cancer stem cell hypothesis is that cancers themselves have a small number of cells that have stem cell properties. And those are the cancer stem cells that are driving the cancer. And that's what I'm going to talk about today. Now let's talk about what stem cells are, and this also relates to the overall issue of stem cells and what's the difference between different types of stem cells. A stem cell is actually defined by 2 characteristics. One is that the stem cell can make exact copies of itself. Well, you could say, don't all your cells divide and make exact copies of themselves? The answer is no. If they're not a stem cell, when a cell divides, the daughter cells begin to change and are different than the parent cell. So in a sense, stem cells are immortal cells because they make exact copies of themselves. But in addition to making exact copies of themselves, they also can morph or change into all other kinds of different types of cells. So they have these 2 properties, and that's what defines a stem cell. Now, there are actually different types of stem cells. The one that you hear the most about, which is actually tremendously important and exciting, are embryonic stem cells because they're found in embryos. And they're tremendously exciting because these are the cells that can morph or change into any other cell in the body. So that they have tremendous potential for to heal regenerative disease, neurodegenerative diseases, diabetes, and those types of diseases. Of course, the reason that they're controversial is, at least of now, to get pure embryonic stem cells, one has to get them from embryos. In addition to embryonic stem cells, there are another type of stem cell. This is the stem cell that are found in adult tissues. Now, the embryonic stem cells actually travel when you're forming as an infant, an embryo. The embryonic stem cells travel to other organs where they then become adult stem cells. And the difference is that an embryonic stem cell can change into any other type of cell, whereas an adult stem cell is restricted in what it can do. So what I'm going to talk about today is breast stem cells. A breast stem cell can make all the cells in the breast, but they can't make the cells in the lung or the colon or in the brain because it's restricted to only be able to make the cells in that organ. So there are two fundamentally different types of stem cells, the adult stem cell and the embryonic stem cell. Now this is kind of a schematic diagram of what actually happens in the human breast, and I'm going to show this because you'll see that this is really important to understand this whole topic of cancer and cancer stem cells and why this is so important. Oh no. Okay. How about if I talk here? You can't see through me though? No. Okay. How about here? Okay. So a stem cell again is defined by a cell that can give exact copies of itself, as you can see here, but it can also make the other cells in the organ. And in the human breast, the other kinds of cells that are made are cells that line the ducts, called ductal epithelial cells. These cells that make the milk when a baby nurses, they're called alveolar cells, and these cells that actually squeeze the milk out of the ducts. And a breast stem cell, then, is a cell that can make all of these cells. Now, most of human breast cancers are what we call infiltrating ductal cell epithelial cells, so it used to be thought they came from here. But what we now know is breast cancers actually originate from these much more primitive stem cells here. And the key of cancer formation in the breast, and we think all other cancers, is that in normal organs, this process of self-renewal, or making copies of itself, is very, very highly regulated. What happens in cancers is that this process goes out of control and starts expanding these stem cells when they should be regulated, and that eventually leads to cancers. Well, all of this was interesting as theory. How do we actually go about proving this and studying these cells? So almost a decade ago, we started studies to see if we could actually isolate and find a human stem cell from the human breast. And this is actually an example. We've actually then been able to identify what we call markers or proteins that are in these stem cells. And this is the end of a human breast here, and this is a marker of a human stem cell. You can see the stem cell is sitting here, but if you didn't have a marker, you really wouldn't know that. And we've now figured out ways of taking these stem cells, and we can actually grow them in petri dishes in the laboratory and start studying what makes them tick. So we can actually take a single cell, and this actually shows a different kind of cell that makes these little glands that fill up with human milk. But from a single cell, we can grow in a little dish now a small human breast. And it's really quite remarkable because it's a breast stem cell. And you actually treat this with milk hormones, they fill up with human milk. We also have figured out ways of actually growing human breasts in a mouse. So what we do is we take the human stem cell and we take the other cells from the human breast and we kind of put them in the mouse. And then when we take a single stem cell, it fills up the mouse breast with human breast cells and goes right up to the edge of the fat layer here. So you could see that the human cells recognize the human environment here, but they can only grow this far. Creating these models really has led us to the next stages of figuring out what goes on in cancer, because now we can actually isolate stem cells and grow breasts in mice and in a dish, and we can examine what happens that goes wrong in these stem cells when cancers actually form. Now, we know that cancers don't develop overnight. Matter of fact, we know that it takes many years for cancers to develop. And in breast cancer, the easiest way to time this, and it's a very unfortunate kind of human experiment, is when women who were exposed to atomic bomb radiation in Hiroshima and Nagasaki, they get breast cancer 25 or 30 years later, particularly if they were adolescent girls when they were exposed to radiation. So the initial mutations that start the cancer start here, and it goes through this process, and the cancer forms, and then the deadly part of the cancer is when it spreads or metastasizes. This can take 20 or 25 years. Well, the only cells that survive that long in the breast are the stem cells, because the other cells grow during pregnancy, but after a woman breastfeeds her baby, the other cells actually die. And the only cells that are left are the stem cells, and they're the ones that build up the mutations. Now, let's get back to where cancers really come from. The conventional model of cancer, again, is that any cell could become cancerous. So that here's a duct in a breast, and any cell here, if it had the right mutations, became a cancer. But the stem cell model is very different. It says there are only a select number of cells here that can become cancers, and when they do, the cancer itself only has a small number of cells that are the cancer stem cells. How do you distinguish those two? Well, Michael Clark and I performed this experiment which kind of started this whole field of stem cells in solid cancers. It had been shown that in human leukemias by John Dick's group in Canada that it looked like they had these cells that were driving the cancer. But what we found is we took human breast cancers now and we used markers that identified those stem cells in the normal breast, just like I showed you before. And we put them in these mice that could grow these human cancers. And what we found was that these small number of cells in the cancer, and it's about 1 to 5% of the cells in the cancer, you take as little as 200 cells, put it in the mouse, and it gets a breast cancer, as you can see here. If you take the other 95% of the cells in the cancer, and you put even 100 times more of them, 20,000 cells, there's no cancer over here, even though these cells make up most of the cancer. And when we looked at these cells, it was actually very surprising what they look like. These turn out to be the so-called cancer stem cells because even a small number of these make these cancers in the mice, whereas these cells, which look very bizarre looking, look much more malignant, actually make no cancers. These are only 1 to 5% of the cells in the cancer. The problem is that almost all of cancer research is concentrated on these cells because they form the majority of the cancer. And we think that that's led us along a lot of blind alleys because it turns out that those are actually dead-end cells anyway. And I'll show you later, those are the cells that we're killing with chemotherapy. The cells we really want to kill are these cells. This just shows if you actually even look at the percent of stem cells in a cancer and look at how well women do. This is actually two from our group in Michigan and our colleagues in France that are looking at this. We just separate these in two categories, and this is the survival of women, and they have low stem cell content or high stem cell content. They die at a much higher rate if they have more stem cells because that's actually driving the cancer. And we can actually follow these stem cells now because we have markers. And I told you that the cancer takes 20 years to develop along here. So we can take biopsies along this continuum and actually look at the stem cells. And I want to show you this one, which is particularly interesting. This is a kind of early breast cancer called ductal carcinoma in situ. This is what happens immediately before the cancer becomes dangerous and can invade into tissue. And what we see is when we stain with a stem cell marker, the stem cells are in red. And this is a duct that's now filling up with cancer cells. Notice that the stem cells are not distributed uniformly, but are right on the outside edge here where they're talking to these other cells, just like they do in that mouse model. And this is the first area of invasion. And notice the stem cells are actually invading out into the tissue. This suggested to us that the deadly part of cancer, which is metastasis, must be mediated by these stem cells because they're like malignant seeds that can go out and spread to other organs. So in order to prove that, we did the following experiment. You all know how fireflies, how they light up in the dark. I guess in Washington they're fireflies in the summer. We have them in Michigan. And they light up because they put a chemical in their tail that makes them glow in the dark. So what we're able to do is to take that molecule and actually genetically engineer it into cancer cells so they glow in the dark, and you can put them into a mouse then and follow where the cancer cells spread in the mouse because they give off light. And even through the wall of the mouse, you have such a sensitive camera, you can see the light that's given off. So that's what's done in this experiment. The difference is you take the cancer, you make all the cells emit light, but then we separate the stem cells from the other cells, and we inject them right into the bloodstream of the mouse. And this shows one example here. These are the stem cells. This is a stem cell marker. And this is a particular kind of breast cancer. It spreads to the bone. You could see it lighting up over here. And this is the non-stem cells. From the same tumor, only the stem cells form the spread in the bone. This one, just like it does, unfortunately, in some of our patients, spreads to the lung and to the liver. And here you could see the stem cells going to the lung and the liver, but the non-stem cells don't form any over here. So what this is telling us is that stem cells are not only critical to cancer growth, but to the spread or metastasis, the deadly part of cancer. And this is a schematic of that. You see in yellow the stem cell, and you can see that even if you had a tiny amount here that spread to this place, if it didn't have the yellow, it wouldn't form a metastasis. But if you have a stem cell, it actually can form a metastasis over here. Now, sometimes we have a very unfortunate circumstance where we have women that we followed for 10, 15 years, and we all think they're cured, and all of a sudden the cancer comes back, like in the lung. How could that happen after 15 years? It turns out when you look at the genes in that cancer 15 years later, they're identical to the one 15 years before, so it's not a new cancer. So what we think happens, we call that tumor dormancy, is that the stem cells spread at the early times before the cancer is actually removed, and they just sit there in a dormant state. And what we're studying now, well, what are the signals that essentially wake up or revive these dormant cancer stem cells 15 years later? Because we think we have to figure out what those are and also, of course, how to eliminate those cells. Now, you've all heard about hereditary breast cancers. These are caused by, the majority of them, by women who have in their families mutations in the genes BRCA1 or BRCA2 for breast cancer 1 and 2. Matter of fact, if you look at the incidence of breast cancer in the general population in women, as women get older, they have a higher chance of getting breast cancer. In the general population, you can see 12%, 1 in 8 women are going to get breast cancer. So it's a terribly large problem in this country. But the women who are unfortunate enough to inherit a mutation in the BRCA1 gene, you see in red what their chance of getting breast cancer, more than 85%. They also have a 30% risk of getting cancer of the ovary. Now, until recently, it was thought that the way this gene caused cancer was that when it went awry, that you couldn't repair mutations in your DNA. It's kind of like a spell checker. But that really doesn't explain why women who have this mutation don't get all kinds of cancer. They only get breast and ovarian cancer. The clue to us was that they get a very specific form of breast cancer that very much resembles what we see in breast stem cells. So that led us to do the following kind of experiment. We actually did our genetic models in the mice, and we knocked out this gene, the BRCA1 gene, just like it happens in patients. So this is what happens when you just grow these cells in mice. They form these little ducts. And some of these express this marker called estrogen receptor, which is what makes breast cells sensitive to the hormone estrogen. Women who have BRCA1 mutations, their cancers aren't sensitive to estrogen. And indeed, when we knocked out this gene and put this in the mouse, what happened is every cell has the stem cell marker here. It looked like the stem cells expanded in the mouse, and none of these cells have estrogen receptor. Well, what does this really have to do with real women with BRCA1 mutation? Many women who carry this mutation, and you could detect it from a blood test, decide to have their breast removed, prophylactic mastectomy, because the risk is just so high they don't want to deal with such a big risk. So they consented us to look at some of their tissue when they took out their breast. And what we found is this is a normal breast lobule. This is the receptors for estrogen. And in red are stem cells. You hardly see any stem cells. Notice this lobule is all red. It's got all stem cells in it. And what we've actually proven, as I'll just show you on the next slide, is that the reason for that is that every woman inherits 2 copies of each gene. When they have a BRCA1 mutation in the family, they have 1 normal copy and 1 abnormal copy, and that's enough to make a normal lobule. So this lobule, they must have lost the normal copy. And indeed, we've actually proven that now, and this is just a way of looking by sequencing the gene. If you look at where they don't have stem cells, they have the 2 copies. You can see it very well here. But here they have stem cells that have expanded in the breast. They've lost the normal copy right over here. So hereditary breast cancer is actually formed because you lose the gene that regulates the stem cells, and the stem cells start to expand. What about all these other women who get breast cancer who don't have it in their family? What's the relation of that in stem cells? Well, 2 very important genes that regulate other breast cancers that are not hereditary are the so-called HER2 gene, which you've heard about because it makes cancers more aggressive. About 20% of women have too much of this gene. But we have good medicines now called Herceptin, which can block this and have been really a major advance in breast cancer treatment, treating women who have HER2 amplification or too much HER2 with Herceptin. And this is another gene that is lost in about 40% of breast cancers. And sure enough, what we found is when we measured the stem cells in cancers where we knock out the PTEN gene or overexpress the HER2 gene, the stem cell number actually goes up. So what this is telling us is that although we always thought that there was no real relationship between the hereditary breast cancers and the more common sporadic breast cancers, it turns out that they're actually very similar from a biological point of view. There are different genes involved, but both of them involve genes that are dysregulating the stem cells. So you get too many of these stem cells, and that's what leads to a cancer. Now, I want to talk about the embryonic stem cell. What was really a shocking thing was just found over the last year by two groups, one at Stanford and one at Harvard. This is the Harvard study. What they did is looked at these stem cells in cancers and say, well, do they really look like in the genes that they express the same as the normal tissue stem cells? So, for instance, does the breast cancer stem cell really express the same genes as the normal breast stem cell? How could it be? If it is, why does it reproduce in such an abnormal way? And the shocking finding was that what happens is that the adult stem cell begins to express the programs that are expressed in the embryonic stem cell. That is the cell that is not normally found in an adult body. It's only found in these embryos and the cells that are so controversial because these are the ones that can form any organ in the body. You can see that women whose cancer stem cells actually express the embryonic stem cell program all die very quickly compared to ones who don't over here. What this is telling us is that we really want to understand what makes these stem cells divide. We have to understand embryonic stem cells. And we have to look at the whole spectrum of embryonic stem cells and adult stem cells, normal and malignant, to understand what's really going on. Now, finally, let me talk about therapy. This is obviously very important. And I get back to what I started with. Why haven't we done better at treating cancer? Well, the cancer stem cell model provides a good explanation for that. If you have a cancer stem cell shown in yellow, and you have treatments like chemotherapy that kill 95% of these cells, just like leaving the roots on a plant, you still have the cancer stem cell and it can regenerate the tumor. What you really need to do is knock out the cancer stem cell. In an interesting model, does it really occur? Well, our colleague Ginny Chang down at Baylor in Texas reported this year the following. What she did is to look at women who were getting chemotherapy for breast cancer but had very large cancers, so that you give chemotherapy first before you actually take the cancer out. What that allowed her to do was to measure the stem cells in the cancer both before and after the chemotherapy was done. The chemotherapy, after all, shrinks the cancer down, sometimes dramatically. And she used the markers that we had determined on stem cells. So this is the percent of stem cells in the cancer before chemotherapy. And after chemotherapy, it goes up because they're completely resistant to the treatment. We've actually discovered it's even worse than that, because when you give chemotherapy, it turns out that the cells that you kill release growth factors that stimulate the cancer stem cell. And the reason that they do that, it's like a wounded tissue. When your tissues become wounded, they secrete growth factors that signal the normal stem cells in that tissue to regenerate the organ. The same things are happening in cancer, and that's why we haven't been able to cure more cancers. So what this is telling us is that the way we've measured success of our clinical treatments, we think, has been very flawed. Because what we've done is we've measured the ability of agents to shrink down cancers. But if cancers are 95 or 99% not stem cells, shrinking a cancer down by 95% may have no value. And a matter of fact, for most diseases, including breast cancer, shrinking cancers down does not make patients live longer. There's not a good correlation between the two. And our clinical trials are even designed that way, and the FDA approves many drugs by what they call the response rate, which is shrinking cancers down. We think it's the wrong endpoint. And we're in a lot of discussions now about how should we change our paradigms of thinking so we could get endpoints that will be measuring cancer stem cells. Our challenge is to develop therapies that can target the cancer stem cell but spare normal stem cells that you need to live. So you need stem cells in your bone marrow or in your intestine. You can't knock those out. So we're going after particular genes that we think may be different in the cancer stem cell from the normal stem cell, and that's where this embryonic stem cell research is so valuable, because it's telling us what the differences are between those. This is just one example. We're actually now in clinical trials with the first one for breast cancer, a particular agent developed by Merck that was actually not even developed for cancer, it was developed for Alzheimer's disease. But it turned out that the pathway called Notch, which was involved in Alzheimer's disease, is exactly the one that's making the cancer stem cells work. So we've formed a collaboration with our colleagues at Baylor and at Harvard now to actually do clinical trials to target cancer stem cells. Now, I only talked about breast cancer today, but since the breast cancer stem cells were discovered now about 6 years ago, very rapidly, this all has been filled in, and all of these cancers now have been shown to have cancer stem cells. And what's very exciting is that the markers and the actual targets on these cells may be shared between different organs. So the idea that we were going to develop treatments that would just be for breast cancer or colon cancer, and the way we fund a lot of things in our advocacy groups that are very much targeted along one line or another line of disease, what we're learning in science now is taking us in a different direction. what we're learning is that what we learn from one disease really informs and tells us about another disease, and some of the treatments that may work very well for one cancer may actually work for another cancer too if those stem cells happen to have the same pathway that's dysregulated. The excitement, just as shown, one of our leading clinical journals just a couple of months ago had the whole issue was on cancer stem cells. So now it's actually caught on. People were very skeptical in the very beginning of this because there was so much against the paradigm of the way we were going, and now I think we're going to see lots of clinical trials over the next several years, and funding from the National Cancer Institute is absolutely critical for this, and the Cancer Institute is actually putting targeted money into cancer stem cell research. And the type of research that we feel is really critical is what we call translational research, because we need the basic research to understand what to even target. Without the basic research, we really can't go anywhere. The difference is kind of going from the old way of treating cancer, which was completely trial and error, very empirical. We had no understanding what was going on, so we kind of attacked it with chemotherapy, which is like a sledgehammer. And we need to go from there from very carefully targeted molecular therapies that are targeting the right pathways in the right cells, that is the stem cells. For those of you who said Michigan never has sun, look, see, there's a rainbow in there too. So I hope that I gave you just a flavor for why we're so excited about this field, and I'd be happy to take any questions about this. And we also have other experts in the audience on embryonic stem cell research too, and we'd be glad to take any questions.
May I say a word? Okay. I'm Representative Holt from New Jersey, one of the co-chairs of this caucus, and I wasn't here to introduce you.
Okay.
I apologize for that. I'm pleased that I got here for part of the presentation, and I hope you all understand. I think this is maybe a particularly good example of why this caucus exists and why we hold these meetings. I think the line from the research to therapy is at least suggested for you, if not filled in. And this is obviously directed at helping people, reducing morbidity and mortality. And so I thank you for that. It's a particularly good presentation. If I may begin with a question of somewhat amateurish question. Going back a decade or two, before you had the markers for the stem cells, in fact, before you had recognized the stem cells, were these 1% of cells identified as I think you said 1 or a few percent that are stem cells. Were they recognized as different from the other cells? Or was it only when you got a marker that you began to recognize them as different? And if that's so, how did you develop the marker? I mean, how did you know to develop something to mark something that you hadn't seen or didn't even know was there?
So it's a very good question.
And I'm leading up to another question.
Well, one of the things we knew was that it was very inefficient when you try to take a cancer and put it into a mouse, say. You had to put in 20, 30,000 cells in order to get a cancer. But the conventional wisdom was that they're all the same. It's just that it's a very inefficient process to grow it. Until we had the markers, there was no way to say for sure whether there really was a subset that was specific that was really driving this. The key, though, was to be able to have a good animal model where we could actually test this. And it turns out that the markers that we use to pick out the cancer stem cells are also markers that pick out normal stem cells. So it's not as if you just have to pick any marker.
You appropriated this from other stem cell research.
Yes, exactly.
That others had done probably with embryonic stem cells.
Other stem cell research, right, because other stem cell research is absolutely critical. Matter of fact, all of the pathways that we attack on the cancer stem cells, we start looking for them by ones that are being identified in the embryonic stem cells. And particularly now with this recent research that I showed you before, that the difference between an adult stem cell and a cancer stem cell is largely due to activation of embryonic stem cell genes.
Okay, so if I may follow then, you now have a marker that will chemically recognize these cells and tag them for you. Does that suggest a therapeutic approach? In other words, if there's something that will see these stem cells, latch onto it, so that you can see them or measure them, why not attach a deadly drug to that same
Well, that's exactly what's being done. So the first marker that I showed you that was called CD44, there are already 3 groups that have developed specific antibodies against that and are now showed that in animals with cancer you can hit the cancer stem cells. Just as you said, you can come work in my lab, you have good ideas. So that's actually what's going on. A number of companies are approaching it exactly the way you say right now. They've looked for targets on these stem cells and they're making ways to actually target them and bring toxins and things right into those cells. What we're learning, though, is that the cancer stem cells are very resistant to these therapies. And when you think of it, that your stem cells are so important in the body that they've evolved ways of protecting themselves. So when we give a patient chemotherapy, for instance, the blood counts go down. But unless you give huge doses, they come back up. The reason for that is that the more differentiated blood cells are all killed by the chemotherapy, but the stem cells in the bone marrow are more resistant, and that's why they come back. Same as in cancer. The other cells are much more sensitive. You give chemotherapy, they die quickly, but the stem cells are very resistant. So in addition to making targets on the cells, we're trying to figure out why are they so resistant and how can we overcome that resistance. Yeah, question. Yeah.
Are cancer stem, I have 2 questions, but one's really short. Are cancer stem cells equally resistant to radiation? And then that's the fast one. And then the other one is in your list of cancers that you put up here at the end, there were none of the HPV-related ones like cervical, anal, penile. Does the stem cell thing, is that irrelevant to them, or has just nobody looked at them yet, or what?
So the first question is that stem cells are also resistant to radiation. And this has been shown actually very well now in 2 cancers, one brain cancers, and the other is breast cancer. And the brain cancer, we treat it as a primary therapy is radiation. And this shows another reason why this shrinks down, but that we don't cure any patients with brain cancers because the stem cells are resistant to it. As to HPV cancers, it's a very interesting question. It turns out that Dr. John Niederhuber, who's the head of the National Cancer Institute, is working on that himself in the laboratory. And what he's found is that the viruses that infect cervical cancer cells preferentially infect the stem cells in the cervix. And they hide in the cervical stem cells, and that's probably how they actually cause cervical cancer. So it's going to be a very important issue of how these viruses actually infect stem cells. It turns out that if they infect other cells that are not stem cells, they may do that too, but those cells die anyway. It's only the stem cells that keep passing on the defects in the genetic information to the next cell. So those are the key cells that get infected and become cancerous. Yes?
Women that test positive for the BRCA1 have their breasts removed. What is their survival chances, and what is the next step that they should take if they go ahead and have the breast removed before the cancer is detected but the BRCA1 is positive?
So women who have the breast removed have a 95% protection then against breast cancer. And the reason that they still have a 5% risk, which is actually then even lower than the general population, which is a 1 in 8 risk, is because even when they remove the breast, there's a tiny amount of breast cells sometimes still on the chest wall. But 95% of the tissue is removed, so it's actually been looked at now. So there are some recurrence, but it pretty much protects a woman if she hasn't gotten the cancer yet, removing the breast. It's just a very drastic thing to have to do. The main problem then is the ovaries, which also have a 20 to 30% risk of ovarian cancer. Some very early studies in that now suggested it may be the same thing, and that's what we thought from the beginning, that BRCA1 also controls ovarian cancer stem cells.
So
well, I mean, normal ovarian stem cells.
Positive response, then you should automatically, if you're aggressive, you would take out the ovaries also to avoid that.
Yes, as a matter of fact, what's recommended now is that, particularly after a woman has had the family and decided she's had all the children she wants, that the ovary should be removed. And because in particular, ovarian cancer is even much harder to detect than breast cancer, and unfortunately when we find it, it's usually too late, and the screening tests just don't work very well. So that's particularly important because it is such a deadly disease.
So a BRCA1 positive tester and removes the breast, and she has a pretty good chance if she takes the ovaries out.
That's right, to live a completely normal life, yes.
Thank you.
Now we hope someday that it won't be necessary to take off the breast, but now there is a risk. We know, for instance, that taking tamoxifen or that taking the hormones reduces the risk in half, but then it goes from 80% to 40%. So it's still a very big risk, but it's certainly much better than doing nothing. And if a woman does not want to have a mastectomy, at least it's another choice to reduce the risk in half.
These
genes, or these cells that mutate into other parts of your bodies and become and metastasize. Do you know anything about when those actually migrate?
It's a very important question, and our ideas of this are really changing very radically because of new research. We used to think that metastasis was a very late thing that happened, that the cancer develops, and once it gets to a certain size, it starts putting cells off. But the new research indicates that metastasis may happen very early in some tumors, and that the chance that a cancer metastasizes is more related to what particular genetic mutations are in the cancer, and we think how many cancer stem cells there are, than how late the cancer is. And in a way, that's pretty frightening because it means that early detection can certainly help, but it may not be the total answer because no matter how small you find it, it may be that sometimes some of the cells may have spread already.
And they're just waiting there?
Well, we're trying to figure that out because the other thing that's all of our ideas in cancer research are changing so quickly and so dramatically. We used to think that the cancers, when they spread, just grew on their own. But now what we're finding out is that cancer is really a disease of the whole body. And there are factors that circulate even between cancer cells and the metastasis, and they signal each other. And that may be the reason why when you remove the primary cancer, sometimes you don't get metastasis. Even though there may be tiny ones somewhere else, it may take away the mother home here that's signaling them to grow, and maybe these women may never develop metastasis. We have to figure out why the small number that develop metastasis 15 years later, why they were there all along, why do they come out of dormancy after all of that time? Now, women in my clinic have told me, I have a number of women like this, and for many years, most of them come up with a story. And this is all very anecdotal. What I'm saying now is not scientific at all. But they would tell me I'm in an auto accident, and all of a sudden my cancer came back. And I would always say, there's no relation. We don't understand, there's no relation to it. But what we're finding now is that these signals that signal the dormant stem cell to come back into cycle again are in the same genes and the same signals that may be involved in tissue injury. So actually, I think that there is more of a connection than we ever knew, and it's because of what I said before, that the body is very well situated to heal big injuries. So for instance, when you have a big injury, you get stem cells that come from your bone marrow. They're called mesenchymal stem cells. They travel from the bone marrow to the wounded tissue, and they make growth factors that heal the tissue. What we're learning now is that cancers do the same thing. The cancer sends out a signal. It gets these mesenchymal stem cells from the bone marrow that come and tell the cancer to grow and make the cancer grow. So the cancer has usurped some of the same mechanisms that we're getting in tissue injury. And it shows, again, in research that you have to have a broad understanding and a broad view, because what you learn in an area may actually affect another area that's completely different from it. In this case, inflammation in cancer. I told you before, like Alzheimer's and cancer also, they may be actually very related in their basic research. Yes.
We often talk about killing the cancer cells, but presumably because the breast develops naturally, is it possible to revert the cells to their healthy state?
Yeah, so that's another interesting area of research, and the idea is, can you take a cancer stem cell and push it to say differentiate so it loses the ability to make copies of itself? And that's another area of research. It's a very important one. A similar kind of thing has actually been done in certain kinds of leukemias where the retinoic acid, the vitamin, they have a particular gene problem that blocks the stem cell so it can't differentiate. But if you give very high doses of vitamin A, it actually treats those patients because it pushes their stem cells to differentiate. So the idea is a very good one, and we have already a good model where it could work. We have to figure out how to do it. Yes?
So a lot of times when you see media reports about stem cells, they're actually talking about injecting stem cells as the actual treatment. If I understand correctly, that's not at all what you're talking about here. You're actually using the stem cells to learn things to develop non-stem cell treatments for the cancer. Is that right?
That's right. I mean, what it's telling you is that the biology of stem cells, including embryonic stem cells and adult stem cells, is critical. It regulates basically everything that goes on in your body. If you understand how to control that, you may be able to use them for tissue regeneration. But you also can understand when that goes wrong, it causes cancer, so it may tell us how to attack the cancer too. Even though they're two very different problems, it's a continuum of understanding what's really going on in the stem cell. Yes?
Can you discuss a little bit the necessity of embryonic stem cells to do this sort of research? I know you brushed upon it a little bit, but what stage was their necessary presence, and do it without embryos?
I think the embryonic stem cells are critical to understanding what's really going on, because particularly now that we know that cancer stem cells are somewhere in the middle of embryonic stem cells and tissue stem cells, unless we're able to study the embryonic stem cells, I think we'll really be impaired in our ability to understand what's going on in the cancer stem cells. Now, this is different than what we're saying of the other reason to use them for regenerative diseases. That's another very important reason, but in cancer, we need them to understand what's going on in the cancer cell.
So the embryo must be fertilized in order to retrieve the stem cells, is that?
That's right, because the embryonic stem cell is the only one that expresses all of this program of genes, a number of which are driving the cancer. And how they shut off and why don't get more cancers is really critical too, because develop in the normal pathways. These things must get shut off, and somehow they get turned on again when the cancer forms. They get turned on in the adult stem cells.
And do the genetics of the sperm affect the embryos as far as comparing the research?
Well, I don't know that it's so much the genetics of the sperm itself. It's the genetics of the stem cell. It's the embryonic stem cell itself that's so critical, because the secrets of what's going on in the cancer are locked inside the embryonic stem cell. So.
Hypothetical. If you have a brother and a sister, siblings, they have identical DNA. One gets breast cancer, the female gets breast cancer, the male gets leukemia. Do you see a correlation there?
Yeah, so it gets back to, we actually are learning that cancers have actually a lot in common, and some of the genes that go wrong in one cancer, BRCA1 may not be a good example because that's breast and ovary, but some of these other genes can affect many different kinds of cancers if they hit one stem cell or another stem cell. But it also means that, again, some of the therapies we develop for one cancer will be useful for another. So in the future, the difference is going to be that you're not going to get treated just based on the type of cancer you have, that if you have breast cancer, you just get this treatment. If you have prostate cancer, you just get this treatment. It'll be that you'll have an analysis of your own individual tumor and the stem cells in your tumor and say, these are the genes that are going wrong in your tumor, and this is what you need to block that gene. It may turn out that it's the same in breast and prostate instead of just being in the different diseases. And that's why in the politics of funding, this becomes very confusing and difficult, because advocacy groups are so oriented towards their own disease side. But scientifically, that's really telling us how limiting that approach is. And a great thing about NIH is that it's a much broader attempt to look across all of these and find these commonalities.
So that would explain why family members develop different kinds of cancers?
Yeah, as a matter of fact, there are a number of genes that we know now, like one called P53, that if it goes wrong, it causes a whole lot of different kind of cancers in different family members, but it's the same gene. Yes?
Follow up on your last statement. Is this area of work being funded by the NIH? And is there any way to quantify how the work is being held back by the limited funding of the NIH over the last 5 years?
The vast majority of this work is being funded by NIH. There are other organizations and foundations that are just getting in to fund this, but all of the real basic research that showed how stem cells work and cancer stem cells, that was all funded by NIH. And it's really is a problem with the limitation of funding. Not only, it's not so much of a problem now for a few established labs, like our lab can get money from NIH, but what's really frustrating is young people who are really bright, who want to get into this area, are reluctant to do this because it's so hard to get a grant from the NIH now. Only 12 or 13% of grants that are being put in that they say are worthy of funding are getting funding. And some of the really bright young people say, I don't even have a chance. Why should I do this? I'll go into business. And we could lose a whole generation of researchers that we really need to carry this kind of thing forward.
Let me add another word about that, because I was talking earlier about the reason for this this caucus and these meetings. Another way of saying what Dr. Wisha said is, as I understand it, it is now typical that the age that a person, an investigator, first becomes the primary investigator is sometime in her or his 40s, I believe. Is that right?
Right, it's going up all the time.
You know, think about your career. You know, you don't want to wait that long to make partner at the law firm or to make partner at the financial firm or whatever. Well, It's the other reason we're here. So, I mean, it clearly shows that there is more talent than there is funding. And Lord knows there's a need here. I mean, we know about the incidence of cancer and these other things. So, you know, take it back to your office that there is a critical role for federal support here. But I would draw it a little more broadly than that. Dr. Wisha didn't say it, but I will. You and the interest groups, the advocacy groups for individual ailments who come to see you should lobby just as hard for funding for the National Science Foundation and NASA and the Department of Energy so that we will have the methodology, the instrumentation, and the scientists who will be doing the NIH research. They are funded at a much lower level. I mean, a fraction, you know, a 5, 10, maybe 20% of what NIH is. So I'm not trying to say that don't fund NIH. We need to increase funding there for those reasons. But we need to make, and please take this back to your bosses, we need more funding in the basic sciences. That includes physics, that includes engineering, it includes chemistry, not just the medical sciences, so that we can do this research that's necessary for reducing morbidity and mortality. So forgive me for stepping on your message here, but and I'm going to have to excuse myself to vote. But thank you for a very good presentation.
Thank you.
Dr. Wisha, I spoke to Dr. Rosenberg about a week ago, and I think he told me 1 out of every 3 people will get a form of invasive cancer, correct?
It's even higher in men. In women, it's 1 of 3, and men, it's 1 of 2.
Well, somebody needs to tell Representative Holt that, because that takes it up even higher as far as we need more medical research money. So,
Any numbers on how many people developing more than one primary cancer?
Yeah, actually, it's quite common now, and because of that, we have major new initiatives in what we call cancer survivorship, which is what to do with people who have had cancer. How should we follow them? Because they are at a much higher risk of getting a different kind of cancer. And we think it's because of this idea of these same genes that are in there. And we probably need to do surveillance and things like colonoscopies, you know, mammographies at different rates and following people who have had other kinds of cancers. But it's an important area now of research. Okay, well, thank you all very much.